With ahead of time to go live at 1 p.m. today, but I want to make sure that the settings work because a couple people weren't able to see it when I expected last time. So if that doesn't show up for you, let me know and I'll get it in there and fix it as fast as possible. Other requests we had were for note taking, posting the slides with that red text filled in after class. Got a lot of back-to-back classes this afternoon, but I will have those up for the lectures we covered definitely by Saturday afternoon, but hopefully I'll find some time this evening to get those up for you. And finally, to sort of summarize some of the stuff that we'll be doing today and get us ready for next week, I'm going to put up another assignment where you kind of label and explain a little bit, looking at a picture of the synapse. Again, hopefully my afternoon lab won't take as long as I think it might, in which case I'll go up this evening, but otherwise definitely by Saturday. So that's what's coming up for you. So now we're going to pick up where we left off. So we're talking about synapses. So synapses are where we have neurons talking to each other. So we have an action potential happening in one neuron, our first neuron, which we're going to call the presynaptic neuron. So presynaptic before the synapse. And it's going to send a signal across to the postsynaptic neuron. So the neuron after the synapse. Now, when we left off last time, we mentioned that there are two different big types of synapses that we could focus on. We went over the less common one last time, which was the electrical synapse. So in the electrical synapse, we have gap junctions connecting our presynaptic and postsynaptic neuron. And a gap junction really just lets anything that's in the cytoplasm flow directly through one cell into the other cell. So even though they're technically separate cells, we kind of think of them as one piece. So in those electrical synapses, that action potential with its ions can kind of just flow directly into the next neuron. We don't really have anything more to worry about when we're thinking about those electrical synapses. So I know you may have noticed every time we say the word electrical, really we're thinking about neurons, or sorry, we're thinking about ions. And now we're going to talk about our chemical synapses, where we're going to spend a lot more time. Our chemical synapses are going to be where we're thinking about like neurotransmitters, so chemicals out in the brain, out in the body. Those chemicals being received by other cells that then in turn react to those signals. Right. So as we're thinking about these connections, thinking about first what the synapse actually is, like where it is, what it looks like. So we are looking at a connection between one neuron, so our presynaptic neuron. See this first one. Okay, so one neuron here. So that's this over here. So we've zoomed in on that axon terminal at the end. So that little foot process here, that's our axon terminal of the presynaptic neuron. And the next thing we see is our second neuron in the chain. So the second neuron here that we're looking at, that is our postsynaptic neuron. On this postsynaptic neuron, we've zoomed in on the point where we have a synapse, the connection between these two cells. In this case, it kind of looks like we've got an axosomatic connection. So between the axon terminal and the soma or the cell body. Looks just based on this picture like that's what we're seeing. But it's more common that we would actually touch the dendrites, these processes. I suspect that's harder to do. That would be an axodendritic connection. So we just name them for what two parts are touching. So we've got our presynaptic neuron, our postsynaptic neuron, and in between, we have a synaptic cleft. So cleft, it just means like a gap, right? So this synaptic cleft is just this little bitty bit of space between these two neurons, right? So this space, the synaptic cleft, is where we're going to be sending out our chemical messengers, which is what makes this a chemical synapse, which will then be received by our second cell in the chain by that postsynaptic neuron. Okay. So we've got our presynaptic neuron axon terminal up here at the top, our synaptic cleft, the synaptic neuron, and then we can zoom in and see what we see on each of these cells, right? So we've got pretty obviously here in the presynaptic neuron, it's kind of little baggy structures, right? So what we're looking at here is a vesicle. So a vesicle is a section of membrane, right? So a section of membrane forming a little bubble in the cytoplasm. And these little bubbles, specifically these ones near our synapse or our synaptic vesicles, are going to start out full of our neurotransmitters. So the chemicals that will eventually be releasing into that synaptic cleft are here represented by those purple triangles. So those are our neurotransmitters, which are our chemical signal. Well, that chemical signal is eventually going to have to get into that synaptic cleft. So we'll see how that happens in a second. Other structures we see here on this presynaptic neuron. So in our axon and in our axon hillock, when we were thinking about the action potential moving along, we looked at two types of voltage gated channels. We looked at our sodium voltage gated channels that had an activation gate, inactivation gate. We're responsible for opening when we depolarize to that threshold value of -55. When they open up, sodium can rush into the cell, which is what causes that spike up to +30, that spike that is really characteristic of our action potential. And we had our potassium voltage gated channels with just one gate. We don't have to name the gates. Those potassium voltage gated channels were opening up a little bit later, slight delay in that action potential. But once they open up, they're letting potassium run out of the cell. That's why we have our depolarization as sodium is flowing in, and then that repolarization as potassium flows out. In our synapse, we have a new type of channel. We're going to see here that we are finally talking about a different ion. So here we see a voltage gated channel for calcium. So calcium is the Ca2+. So we have a new voltage gated channel. So you should be thinking, we're going to be thinking about membrane potential changes in the synapse and what's going to happen when that change in membrane potential, when the electric charge reaches our axon terminal and touches these voltage gated channels. And we saw that we had this synaptic cleft, right? On the opposite side of the synaptic cleft, on the postsynaptic neuron, we see some different membrane proteins. So on the side that's kind of catching our signal, we see that we have receptors. So receptors are going to receive signals. They are an embedded membrane protein. We're also going to see some enzymes embedded here. So enzymes catalyzing different types of reactions are also part of this postsynaptic. Now, our final membrane protein, just going through this list, we have to jump back to this presynaptic neuron. So we see this reuptake molecule here back on the presynaptic. So we're going to step through what's happening in the synapse. So go ahead and look at the figure, the numbered figure that's on the next slide. It's just that same picture, but it's got some numbers on it. Slide 10 in your copy of the slides. I want you to look at it and figure out, are there any molecules from the presynaptic neuron that are actually entering the postsynaptic neuron during the communication here at the chemical synapse? Is anything coming out of the presynaptic neuron and going into the cytoplasm of that cell? Take another couple seconds. We got 50-50 shot here. Take a look. The answer, we'll go through why this is the case. There's actually not anything that is going all the way through from that presynaptic neuron and into the postsynaptic neuron. So we're going to see all these structures in the chemical synapse because we don't have a gap junction. So we don't have like a tunnel from the presynaptic to the postsynaptic neuron. There's no open door between the cytoplasm of the two cells. So we have to do this sort of passing of the signal. We have to do all this passing of like a ball from membrane protein to membrane protein, from signal to signal, from cascade to cascade. So that's part of why this looks more complicated. That's going to be a real difference between the electrical synapse that we looked at the end of last time and the chemical synapse that we're looking at today. All right. So when we are looking at synaptic communication, thinking about the presynaptic neuron communicating, signaling to our postsynaptic neuron, it's all happening at the end of an action potential. So everything we've been talking about up until this point, we've really been focusing in on that presynaptic neuron. So we've been focusing in on a neuron that's receiving signals from somewhere, building up graded potentials until they sum together, get us to that threshold value of -55 millivolts, at which point we have our voltage gated sodium channels opening up. They're concentrated in the axon hillock right at the beginning of our axon. So we see that membrane potential spike up to +30, first in that axon hillock. And then we see that spike in charge moving down the axon, patch by patch, right? Just like a crowd kind of doing the wave, right? So this is traveling down the axon. Now we're looking at the axon terminal that is at the end of that same cycle. So the first thing that's going to be important to realize when we're thinking about the synapse is that this is passing a signal when that first cell, when that presynaptic neuron has reacted to something. So that presynaptic neuron has something to say. It's telling us about a stimulus. It's telling us about something out in the body or something happening in the brain. So we have this wave of charges coming down the axon and ending up in the axon terminal, right? So we have that action potential. And that depolarization, that maximum depolarization, is a spike to positive 30, right? So we're going to a membrane potential of positive 30 millivolts, okay? And when it gets to the axon terminal, right? The axon terminal is also depolarized, right? So we just have those ions still spreading out here. Okay? So in this axon, we have that membrane potential changing from its rest value. So this affects those voltage-gated calcium channels. So that's our next step. When this wave of depolarization hits and touches one of these voltage-gated calcium channels, that voltage-gated calcium channel opens. We're going to come back to the slide, but I have another question for you before we move forward. Okay. Picture, we see that calcium is entering the cell when that voltage-gated channel opens. Okay, so we want to remember why would calcium enter a cell instead of going out of the cell?
Thank you.
Another couple of seconds. Okay, so half of us are here. Okay, so this is why I have the time to go through specifically that calcium example a little while ago. So that was the one where we had to do that conversion from nanomoles to millimoles when we were looking at the concentration. So for calcium, if we're thinking about equilibrium potential, how that interacts with membrane potential, we basically have the same situation as we do for sodium. So calcium has a really, really high equilibrium potential. It's over 100. Okay? Calcium also has that 2+ charge on it rather than just that single plus up at the top. So it has a different valence. So that also makes the force even stronger on calcium ions. Right? It's got a huge concentration gradient pointing into the cell. All right. And relative to our resting membrane potential, it also had an electrical driving force pointing in. By the time that the action potential gets there, that flips a little, but we've still got to add together that chemical driving force and the electrical driving force. And the end result is that the electrochemical driving force is strongly favoring calcium moving into the cell. So that electrochemical driving force is the force on calcium, on those calcium atoms. Right? So calcium is just going the direction it wants to go when we open up that voltage channel. Concentrations and all those other cellular processes set up a situation where calcium has a preferred direction, and all we had to do was open a channel and allow calcium to flow. And so that voltage gated channel isn't using ATP. This isn't active transport. This is just like facilitated passive transport. This passive transport through a channel. It's going in its favored direction, just following those forces that we've been talking about. I'm talking through that in part so you can just remember that calcium is flowing into cells. We'll see calcium involved when we think about muscle cell contractions, some hard stuff. So it's worth realizing that calcium wants to come into cells. So our action potential gets here, our wave of depolarization. That voltage triggers the opening of this voltage gated calcium channel. It opens up and calcium flows into the cell because that's the direction that calcium wants to go. So once we have calcium in the cytoplasm, there in that neuron, so in that axon terminal, we got calcium in the mix, more calcium. The cell responds. So this really acts as like a trigger for our next step. So we're not going to look at the specific molecules or receptors or whatever involved here. All we need to know for our purposes is that calcium being here, right, in this cell inside, right, this triggers a response in these vesicles, so in these bags of neurotransmitter. Specifically, the change that it's going to create here is that it's going to make those vesicles dock on the membrane and then get secreted. Right? So when we say docking, right, we're really thinking just like pulling into a parking space, right? So we're docking on the membrane, right? So this is my vesicle, right? And so that's the docking. So that was a response to calcium. And when we have a vesicle touch a membrane, right, touch a plasma membrane, that wall of the vesicle is made of plasma membrane. That end plate on this presynaptic neuron, also plasma membrane. That bubble basically bursts, and it bursts out, right? So that bubble comes in, and then it's just folding out, so it bursts, right? Just like a soap bubble merging into it. So that neurotransmitter that we had inside is now suddenly on the outside of the cell. Okay? So this is basically the process of exocytosis. Right? Exocytosis is how we transport large molecules inside a cell to the outside of the cell. Vesicle coming out, docking, so bumping into that plasma membrane, bursting open, and its contents spill out. And so all of that is just a response to calcium being inside the axon terminal. And so all of that, our signal, the calcium's in there because the action potential got here and opened that voltage gated channel. So this is how we get neurotransmitter into the synaptic cleft. So we can see these bubbles have burst, and now we got neurotransmitter floating around. That neurotransmitter doesn't actually go into our postsynaptic cell. It's not going into like a dendrite or into the cell body. It's not getting incorporated physically into the cytoplasm of our postsynaptic neuron. What it's doing instead is it's binding on to a receptor. So a receptor receives signals. So it's got that name. So this receptor is here. So neurotransmitter is in the synaptic cleft. It's diffusing, it's moving around, right? Diffusion is really just jiggling and movement of our molecules, right? Eventually it jiggles and bumps into a receptor, and that receptor is shaped in such a way that they bind together. So our neurotransmitter binds on to that receptor. Then we're going to have a response in the postsynaptic cell. So when it binds onto this receptor, something about the state of that receptor changes, right? So something about its shape changes, and then the postsynaptic cell, because that shape of this protein on its membrane changed, has some type of internal response. Later on, we'll be talking about different types of receptors and therefore different types of cellular responses. Okay. So some kind of response is what we want to know. So that postsynaptic cell, as long as that neurotransmitter is bound to its receptor, long as we have that physical connection, that receptor keeps whatever shape it is that it changed into. So that cell on the opposite side of our synaptic cleft is just doing whatever internal process that caused, okay? And that reaction stops when the neurotransmitter leaves, okay? So when that neurotransmitter leaves our synaptic cleft, right? So leaves out of this space, that's when we stop having this next cell responding. So in order to stop this signal, so to turn it off, we've got to get rid of all these neurotransmitters floating around, okay? So when we say get rid of them, there are two things we do. So two things we can do. And some of this would depend neurotransmitter to neurotransmitter, okay? First thing on our list is that we have this neurotransmitter degrade. So this is why we have this enzyme on our postsynaptic cell. So enzymes catalyze reactions. If make reactions happen, make them happen faster. This enzyme on the postsynaptic cell, the reaction it's catalyzing is breaking that neurotransmitter. As soon as we break the neurotransmitter, it doesn't work the same. So it's not going to bind on to any more of those receptors, not going to keep that response going in the postsynaptic cell. We're just going to have little neurotransmitter bits left, and then they're going to float, right? So they can diffuse out of the synaptic cleft. So that's one way we get neurotransmitter out of our cell, or out of our synaptic cleft. The other thing that can happen with some of our neurotransmitters is that instead of getting broken down and floating out, our original cell, our presynaptic neuron, can pull them back in. So here we have our reuptake molecule here. So it takes it back up. That's why it's reuptake. Okay. So it can take this neurotransmitter that's floating into the synaptic cleft. Okay. Taking this into the synaptic cleft, binds it in, and pull it back into another synaptic vesicle. So basically, this is a mechanism by which we can just reuse neurotransmitters instead of having to make new ones every time. Okay. That neurotransmitter can leave the cell into the synaptic cleft, react with that postsynaptic neuron, react with its receptors, and then the original cell is like, okay, I want it back. Takes it back through this reaction. Pause now for some questions. I'm going to pull up. So I think this was from a while ago, but when we're talking about that 30 millivolt charge of our action potential, depends which voltage channels you would need. So this is when we have those inactivation gates of our voltage gated sodium channels closing, but we're having our voltage gated potassium channels opening at the same time. For our next question, if a neuron receives a signal, this is another neuron. So when we use these words postsynaptic and presynaptic, I think I understand what this means. If I don't answer your question quite right, or don't get to what you were asking, let me know. So we can have a neuron. So let's think in the brain, right? So imagine we had one neuron in a chain, two neurons in a chain, and then like three neurons in a chain. I think this is the sort of situation that we're asking about. So we won't really talk in this class about connections like that. We're usually just going to be focusing on two things. What I would say about this is it would be specific to the synapse. So we would focus on, okay, what is happening at this synapse, and then name a presynaptic and postsynaptic. And then we would think, okay, what is happening at this synapse? And then we would have a presynaptic and a postsynaptic. So yeah, if we had a chain of three like this, but we were still using those terms, right? Yeah, for one synapse, this would be considered our postsynaptic cell. And then for the second synapse, it would be considered our presynaptic cell. So I'll only ask you if it's postsynaptic or presynaptic with reference to a specific synapse. So with reference to like two specific neurons. Takes us to kind of the timing perspective here. So there is a little bit of time between when our action potential gets to our axon terminal. So between this number 1, right? And then the change in our postsynaptic membrane potential, right? So this takes a little bit of time here. Varies depending on the synapse, varies depending on neurotransmitters, receptors, all sorts of stuff. Okay. This is mainly caused by how many steps we have to go through, right? So there's a little bit of time because we have this wave of charges coming down the axon, right? They have to trigger. They got to trigger this voltage gated calcium channel. That calcium has to trigger the docking of those vesicles. And then that neurotransmitter has to get released through this like exocytosis process. Then also we have to have our binding on to this receptor and then the cell actually responding, which depending on the receptor type could also take some time. Okay. So that's largely why we have a delay here, right? So sometimes people think that it has to do with like diffusing across this space, but this space is so small that it's not really that travel that takes time. It's the fact that all these internal cellular processes each have their race of speed. So it's that we have to go through all these steps that really take so long. Right? Notice this delay, right, is going to be characteristic of a chemical synapse where we have all these steps, but not really of an electrical synapse where it just had, right, like imagine a sodium ion, right? We had a gap junction, it would have just gone directly through, right? No real steps involved there. So that's why the electrical synapses can work so much faster. So now we're going to be thinking a little bit more in detail about speed at our synapses. Okay. We are still thinking about chemical synapses basically for the rest of forever. So we've left aside our electrical synapses, but chemical synapse to chemical synapse, cell to cell, neurotransmitter to neurotransmitter, we're still going to see variation in how fast neurons can talk to each other, how fast that signal gets transmitted. Okay. So at the chemical synapse, we're going to have different types of receptors. Okay. So we're thinking about these receptors. We're gonna locate them in space, right? So we had our presynaptic neuron, that's axon, it's axon terminal. That's my presynaptic neuron. And then we have our postsynaptic neuron, continuing down and out. Okay. All right, so we're focusing in on the synapse, right? This synapse. Okay. And now we're thinking about the behavior of receptors here in the postsynaptic cell. Okay. So the receptors receive signals, receive neurotransmitters. So receptors are located on our postsynaptic cell, our cell that's trying to figure out how do we respond to this signal from our presynaptic neuron. Okay. So we're thinking about different types of receptors that we could have here, right? So they're not all the same. So our first and simpler type of receptor that we could have on a postsynaptic cell is something called a channel linked receptor. Channel linked receptors are sometimes called ionotropic. So notice this bit ion here, right? So we're going to think these as being very closely related to the movement of ions, right? These channel linked receptors, these ionotropic receptors, what we have happening, we've got a neurotransmitter coming into that synaptic cleft. So the synaptic cleft would be here where we see the extracellular fluid labeled. So this is where we get that signal coming from the other side here. We're thinking two neurons. So that's going to be the same space. It is filled with extracellular fluid. So we're going to start to have some neurotransmitter here being released from our presynaptic cell. So our channel linked receptor has got a spot for that neurotransmitter. So when neurotransmitter is present, it binds onto that spot. It's essentially acting as a ligand or ligand here. So this is a form of ligand-gated channel. So when that neurotransmitter binds into its spot, it just opens a channel, right? So this is a channel protein, but instead of reacting to voltage, it's reacting to neurotransmitters binding to it. Okay. So we open this channel, and then we can have ion movement through that channel. Okay. So ion movement back and forth, depending on specifically what ion we're interested in, what ion is moving through this channel. Relative to our next type of receptor, this creates a pretty fast change in our postsynaptic cell. So we're going to see a relatively fast change in membrane potential. Our second cell in the chain is reacting quickly because all we had to do was open a channel, right? So imagine we open a channel for sodium. Sodium's going to start rushing. Okay. So quickly, we have a response here, but that response ends as soon as that neurotransmitter leaves. Okay? So as soon as this neurotransmitter falls out of that little pocket, that channel closes back up. So it goes back to its original state. Okay? So this response in our postsynaptic neuron is going to be limited just to the time when we have a neurotransmitter physically present in the synaptic cleft and binding onto those channels. That's going to be a difference from our next type of receptor. Okay? So we have a fast change happening in our postsynaptic neuron. That channel is closing as soon as the neurotransmitter is gone. And so that signal is kind of washing away in our second cell. All right. Still in a chemical synapse, you can have a different type of receptor. Still in that second cell, we're still receiving a neurotransmitter, but now we have a more complicated system. So we have a more complicated kind of protein here. So this is still a receptor. So we still have a neurotransmitter hanging out in our synaptic clefts. We still have a receptor. So we still have a protein embedded in the membrane that catches that neurotransmitter. Right? But as you can see, there is more stuff happening in this figure before we have a response. Okay. So we are also going to call these different types of receptors metabotropic receptors. So think metabolism, all these different steps going on in the second part of the cell. Another term for them here are G protein coupled receptors. And that's referring to their behavior and their shape here. Okay. So what we see happening here is that our receptor, that protein that's catching our neurotransmitter, it's not a channel protein, right? So we don't just have simply, first thing, we bind on a neurotransmitter, channel opens, ions flow in or out. Instead, we have this type of protein where it's associated with some molecules on the inside of the cell. Specifically, this is a G protein here. So we've got a receptor coupled to a G protein, which is why this is a G protein coupled receptor. When that neurotransmitter binds on, that G protein breaks apart, and part of it is then going to move across the inside of the plasma membrane. So think of it as like kind of like, it's like kicking a skateboard or something along the inside of this membrane. Okay? So that's what we see happening here. And so we're kicking part of the G protein along the inside of this membrane. And then we're going to have one of two kind of reactions happening involving other membrane proteins. All right, so we'll go through those in a second. But this initial response that we have our metabotropic receptor binding that neurotransmitter that activates this G protein. G protein breaks apart and bits of that G protein go do other stuff. So we actually, up until this point, haven't had any change in the membrane potential of that postsynaptic cell. We're not there yet. But looking at that figure and comparing it to our channel linked receptor, to the ionotropic receptor, I'm going to take a look and figure out which of these things are going to be true. Let's take a look. We're going to focus more on those metabotropic receptors in the next couple bits. So we'll focus in on deep. Okay. So we're still a little unsure about this at this point. Okay. That's fine because we haven't gone through all the steps of that metabotropic receptor. But what's still going to be true, regardless of what type of receptor we have, when we create a potential, a graded potential in that postsynaptic neuron, it's always going to be caused by ions. Okay? So those ions are always what causes a change in voltage. Right? So we are still eventually going to need ions to enter and leave the cell in order to change its charge. That's just part of our definition of graded potentials. So in our ionotropic receptors, it's easy to see how that happens. We just bound on a neurotransmitter, channel opened, ions move in. Done. In our metabotropic receptors, we have an awful lot of steps, but in both those figures on the previous slide that we're going to go and look through the differences between, we do still eventually open some type of channel for ion. Okay? So that G protein is always located on the inside of the cell, activated by binding on a neurotransmitter through a receptor. But the job of that G protein ultimately is going to be to help us open an ion channel somewhere on that membrane, creating a membrane potential, a graded potential in the postsynaptic neuron so that it can decide whether to fire off its own action potential. So we can leave it there for now. Next time we are going to be thinking about those membrane potential in the postsynaptic neuron. We're going to have to think about those ions.